Okay guys, we'll be back to the channel of biotechnologists. We are going to be starting the new series, and it's going to be under immunology, and we are going to start with immunoglobulins. Okay, so the objective of this discussion, at the end of the discussion, we are going to be able to define immunoglobulins. You're going to be able to give the general functions of immunoglobulins and describe the basic structure of these immunoglobulins. So the definition, immunoglobulins are first of all glycoprotein. They are glycoprotein. This is key. This is important. They are glycoprotein molecules that are produced by plasma cells. They are glycoprotein molecules that are produced by plasma cells, and they are produced in response to an immunogen. An immunogen, first of all, is that specific part of antigen that is able to elicit an immune response. So if your body encounters what we call the immunogen, which is able to elicit an immune response, you find that plasma cells are going to release immunoglobulins. And these immunoglobulins are glycoprotein molecules. These immunoglobulins their key function is to function as antibodies. We know that antibodies protect us from diseases. They're able to fight off most of those infections. There are some even which you never even get any kind of symptom, but these antibodies are able to fight these infections. So in the definition here, the key points, immunoglobulins, they are glycoprotein in nature, that is one, produced by plasma cells, and they're only produced in response to an immunogen. And we've seen that an immunogen is that specific type or part of the antigen that is able to elicit an immune response. Not all antigens elicit immune responses, and not all the parts of the antigen elicit immune responses. Okay, basically that is the definition. And these immunoglobulins, their name is derived from the finding that they migrate from globular proteins when antibody containing serum is placed in an electric field. So when an antibody if you put serum which contains an antibody in an electric field, you'll see that these immunoglobulins are going to migrate with the globular proteins. So that's how they got their name, globular proteins, immunoglobulins. They are antibodies, and they migrate with the globular proteins. Therefore, it's obvious they are immunoglobulins. So I hope you understood how their name is derived. Okay, let's continue. Now, the immunoglobulin functions. The function number one is antigen binding. As you shall see their structure, these immunoglobulins are able to bind on one or a few closely related antigens. There are some of these immunoglobulins which bind to only one antigen, and there are others which bind to more than one, but they are closely related. And we see here that the primary function of these antibodies is antigen binding. First of all, if an antigen if the body realizes that an antigen has attacked it, these immunoglobulins are first going to come and bind to the antigen. First of all, if this is very important, if you're fighting someone and if an enemy attacks and you're not able to arrest them and detain them somewhere, they are going to come and do harm. Therefore, these immunoglobulins are very important in that they are going to bind. It's as if they have arrested. So this is like the police doing its job. It will first bind to them, and this is very important in protection of the host. If that antigen was meant to destroy your cells, it's not going to destroy. Okay. Now we also have something we call valency. I think we've come into contact with this. We've interacted around this word in another form, but in immunology, this one basically means the number of antigenic determinants that an individual immunoglobulin can bind. So the valency of antibodies refers to the number of antigenic determinants that an individual molecule can bind. If our immunoglobulin is binding to 1, it means it has a valency of 1. So the valence of all antibodies, at least it should be 2. It should be able to bind to 2 antigenic determinants. It should be having 2 antigenic determinants, or which bind to the antigen. However, in some instances, you find that an antibody has more than 2. So we are going to continue with the function. The second function, the first function we've seen it's binding to the antigens. The second function is effector functions. So after binding to the antigen, things not end there. There are some effector functions which have to continue. Now, it is important to know that the binding of the antibody to an antigen has no direct biological effect. It just binds. However, there should be subsequent actions taking place after the binding. And you should also note that not every immunoglobulin will mediate all effector functions. Effector functions include, now these are the effector functions. There is fixation of complement, which results in lysis of the cells and release of the biologically active molecule. So if we are dealing with bacterial cells that have attacked our bodies, one of the effector functions that takes place is fixing of complement. And this fixing of complement is going to result in lysis of the bacterial cells which have attacked the body. So this is key. Then the other effector function that takes place is binding to various cells like phagocytes. Well, we have several cells in the body which play a very important role in fighting against disease-causing germs or most of these infectious agents. And we have phagocytes. These ones they practically eat the infectious agents. We have platelets, mast cells, basophils. So some of these immunoglobulins are able to bind to several cells like the phagocytes and so on. So when they do that, the binding can activate the cells to carry out phagocytosis. So if your immunoglobulin binds to antigen first of all, then on the other side it binds to these cells, like the phagocytes and the basophils. So this binding can activate the cells to carry out phagocytosis, which phagocytosis is more like eating, cell eating. So This is very essential in the immune response against infection. We've looked at the basic, just the definition and a brief discussion about it. We've looked at the major functions. We are now going to look at the basic structure of immunoglobulins. So, immunoglobulins that we are talking about, how are they structured? How do they look? We are going to look at the structure, but let's first discuss about the structure and what makes the immunoglobulins up. It is important to note that immunoglobulins are built from the same basic units. The same basic units. We have different immunoglobulins. However, they all have the same basic units. They are built using the same basic units. However, these immunoglobulins differ structurally. So these basic units differ in different ways. We shall be looking at that as we go on. Now, the very first component of these immunoglobulins is the heavy and light chains. So these immunoglobulins have chains, and we have 2 heavy and 2 light chains. The chains are 4 in total, and they are composed of 2 identical. The light chains are identical to each other, and the heavy chains are also identical to themselves. Other than the heavy and light chains, we have the disulfide bonds. The disulfide bonds are in 2 forms. We have the interchain disulfide bonds. So here, the heavy and light chains and the 2 heavy chains are held together. Now, these chains are held together by interchain disulfide bonds and by non-covalent interactions. So here we've talked of the 4 chains, and we've seen here that these chains are held together. They don't just they're not just in space like they are held together by disulfide bonds, and specifically the interchain. It's between the chains. Okay, it's between the chains. We shall also see the other one, which is the intra. Intra is in the chain. You understand? So we see that we have the intrachain, the disulfide bonds. Now these ones are within the polypeptide chains. We know that these chains, the heavy and light chains, are made of amino acids, meaning they are proteins, and these chains, they also contain the disulfide bonds, which also give them the shape they have. So we have seen the 2, inter and intra. Inter are between the chains. Intra are within the polypeptide chains. Another third characteristic structure of these immunoglobulins is the variable and constant regions. The variable denoted by V and the constant regions denoted by C. Now here, we see that the heavy and light chains are divided into 2 regions based on the variability in the amino acid sequences. If we have the heavy chain, it is divided by 2, and also the light chain divided by 2, based on the variability of the amino acid sequence. This was observed when the amino acid sequences of the different heavy chains and light chains were compared. So here we have the light chain. It is variable light. Let me change the pointer. There's a pointer. So we have here the variable light, constant light. Then here we have the variable heavy and the constant heavy. So these are basically the variable and the constant regions of the different chain. If we are dealing with the light chain, we have the VL, CL, variable light chain, constant light chain, and then variable heavy chain, constant heavy chain. Another key thing that you have to look at is the hinge region. Now, this is the region at which the arms of the antibody molecule form a Y. We are going to see the structure. Let's just be a bit patient. So there is a hinge region at that point, and this region allows for some degree of flexibility, and that's why it's called a hinge. If we know of a door hinge, it is one which connects the door to its door frame, and there is that flexibility. That's why you're able to open that door, to open and close. So it is why it's called a hinge. So it's the same case here. These arms of the antibody molecule, at that point where they form a Y, there is a hinge, and it allows for some flexibility. So we've looked at the hinge region. We have even the domains. We see that three-dimensional images of these immunoglobulins. When you look at these immunoglobulins at a three-dimensional view, it will show that it is straight. It is rather folded into globular regions, each of which contains an intra-chain disulfide bonds. So these regions are called domains. So when you just look at it from a distance, you may see that they are straight, but they are not. So if we see that these regions, that the polypeptide chains, they have the globular regions, and these regions each contains an intra-chain disulfide bond. So these regions are called the domains. Okay, so we also have the oligosaccharides. You see that these carbohydrates are attached to the CH2 domain in most of these immunoglobulins. However, sometimes you may find that the carbohydrates are not at the CH2 domain. We've looked at the domains, and these are the points when the globular proteins fold due to the intrachain disulfide bond. So sometimes, most times, you find that these carbohydrates are attached to the CH2 domain in most of the immunoglobulins. However, these carbohydrates can also be attached on other locations on the immunoglobulin. So this is what we've been trying to talk about. We have the heavy chain. This is one of the heavy chains. We have the light chain. This is also a heavy chain. The heavy chains are all green, and the light chains are yellow in color. So we've seen that there is a hinge. At this point, where the Y is formed, there is a hinge. We've looked at the different parts. We've looked at the variable and constant. Now, if you see here, you find this is the light chain, and so this is the variable light. This is the one which is on the variable heavy chain. This is the constant. This is the constant. So it means that these sites of the antibody they change. The amino acid sequences change, and this makes them able to bind to different antigens. Because if it was the same, it means there wouldn't be that kind of specificity to the different antigens. Okay. So to summarize what we've discussed, we've looked at what immunoglobulins are. We've described the structure of the immunoglobulins. Also, we also talked of the major functions of these immunoglobulins. So in case there is something you didn't understand, just rewind the video and try to re-understand, write down something. Okay. Some of the guiding questions I managed to get was, number one was define immunoglobulins, and then you outline their general functions. Then using a diagram, you describe the structure of an immunoglobulin. It's specifically immunoglobulin gamma. So these are the guiding questions. Try to answer them and mark yourself. Okay, thanks for watching. Thank you a lot. Please don't forget to like the video. It motivates a lot, and it helps me to make more of these videos. And I wish you all the best in your endeavors, in your biotechnology journey. Bye bye.